Perfect. Well, hello everyone, and I just want to start by saying thank you so much for sparing some time out of your busy day to attend this webinar. I certainly know it's getting very close to the end of the year and that everyone's probably very busy getting everything done before Christmas, as Marta and I were earlier this morning doing some Christmas shopping. So we certainly appreciate you guys making the time for this. For those who don't already know me, my name's Geoff and I'm a Science and Technology Advisor with 10x Genomics. We're going to cover the agenda for today's talk in just a moment, but I just want to quickly say that the purpose of this presentation is really to showcase the new advances to our Chromium single cell portfolio that are not just raising the bar for high quality data, but also driving down costs and expanding the scale of experiments. All right, rather than just introduce myself, I want to showcase the entire local support team that you have at your disposal. It's not just the performance of our product portfolio that we pride ourselves on at 10x Genomics, but also the level of support that we're able to offer our end users. So what does end-to-end support mean? For us here in Australia, it means you have trusted partners to help move your research forward, from experimental consultation right through to consultation around sample prep and even data analysis guidance. So you've got access to tailored training sessions. You can speak directly with 10x Genomics specialists. We're all listed here on the slide. You also have the ability to download a bunch of comprehensive resources. So this is user guides, protocols, how-to videos, a bunch of stuff on software. There is a heap of stuff on our support site. So my take-home message from this slide is that we're here to help in any area that we possibly can in order to make sure that your single cell experiment is a success. So now back to the actual theme for today, which is, of course, maximizing data and minimizing costs with new advancements made across our entire single cell product portfolio over the last several months. So we'll begin with a very high level look at what we're going to learn about in this short webinar today. I'm going to start off by covering our latest low-cost Chromium single cell instrumentation, which is designed for specifically running single cell 3' assays. Obviously a fantastic cost-saving initiative. And if you're new to single cell, this is a great entry point. Next, we'll take a dive into some new analysis capabilities that are enabled through our freely available 10x Genomics Cloud. And finally, we'll of course cover all the latest assay and sample preparation advancements across our single cell product portfolio. Each new innovation I'm going to cover today is carefully designed to bring high quality single cell data that you can trust to more sample types, analytes, and applications, and of course to do all of this at a cheaper price. So we'll get underway by covering our new low-cost instrument. So what you see here is the Chromium instrument. It's used in conjunction with a microfluidic chip to partition single cells and oligo-barcoded gel beads into tiny little droplets, which we call gems. This instrument automates critical steps, which significantly lowers the risk of manual errors compared with instrument-free methods, which in turn provides more reproducible run-to-run results. It also eliminates cumbersome pipetting steps and partitions tens to thousands to even millions of cells in just minutes as part of a 1 to 2 day workflow with a hassle-free sort of 3 to 4 hours of hands-on time. So finally, an investment into this instrument means that you can easily increase the number of single-cell projects as your lab and your research grows. So before we take a look at what's new, let's quickly address the two pre-existing versions of the Chromium instrument that we have. We call these Chromium iX and Chromium X series. They look identical, but with the Chromium X, you can run our new multiplexing kits that I'm going to cover in later slides. These kits are designed to make multiplexing very robust and, of course, to save you money. So we know how important an instrument is in relation to generating the highest quality data, but we also don't ever want to see the need for an instrument to be blocking your ability to do any single cell experiments. So that's why I want to introduce our newest member of the Chromium instrument family, which we call the Chromium XO. This is designed to be a very low-cost version of the instrument, which is capable of exclusively running the single cell 3' assay. However, the instrument itself can actually be easily upgraded to the Chromium X version, on which you can then run the full portfolio of single cell assays that 10x offers. So with this instrument, you can start your single cell journey in your own lab, and then when extra functionality is needed, you can simply upgrade the instrument and the instrument's capabilities at the time. All right, so we have a new low-cost instrument, which of course, again, designed to save you money, but how about on the software side of things? How are we helping to maximize data and minimize costs? Well, in Australia and New Zealand, we now have the 10x Genomics Cloud. So what does this cloud actually do? Within the cloud, you can run Cell Ranger or Space Ranger analysis pipelines to process your raw sequencing data into output files that are ready for biological interpretation. Before the cloud, you actually need to install these Ranger pipelines on a local Linux server to run them, which generally meant then you needed to pay for informatics support just to process and store your own data. So I would highly encourage you to make a free cloud account and start running those Cell Ranger analysis pipelines using a really simple web interface and it will get your data really quickly. But what about maximizing data with the Cell Ranger analysis pipeline? How is this possible? So we continue to iterate and build out features on Cell Ranger, and we're actually up to version 9 of this analysis pipeline. Importantly, this new version now contains automated cell type annotation, which is of course the process of categorizing and assigning cell types to individual cells based on their gene expression profiles, kind of, the whole point of why you're trying to do single cell in the first place. So these annotations are needed for understanding the cellular composition and diversity within a sample, but traditionally this is something that you would do on your own using a third-party application once you'd actually got the data output from the Cell Ranger analysis pipeline. So to put it simply, previously you had to run the pipeline, then annotate cells, but now you can annotate cells as part of the pipeline. So let's look at how this works. You start with getting a 10x Genomics cloud account. And again, that's completely free. Very much encourage that. And within there, you generate a static user-specific token. I believe that's just a once-off thing. And then, of course, you're going to need your raw sequencing data in the format of a FASTQ file. Next, you simply run the Cell Ranger analysis pipeline with the option to annotate your samples. And now all the relevant output files not only contain your single cell data, but the cell types in this data have now been annotated. And while you certainly may want to iterate further to refine your results, we hope this is going to be a great starting point that will not just accelerate your analysis, and we're certainly very excited for you to try it out. So this is just another example of how we're continually investing in further improving your analysis experience with 10x data. Okay, at this point, we've covered off maximizing data and minimizing costs through the new Chromium XO instrument and the 10x Genomics cloud capabilities. But onto the final, and I guess likely most critical section for most people here today, which is our new single cell assay capabilities that we've launched over the past several weeks. There's been a sort of a string of several launches, so we're going to try and condense this all into one message. So to make any progress in unraveling the complexities of biology, you're going to need the right tools at your disposal. You need technologies that provide insights at the fundamental resolution of biology, which is the resolution of a single cell. So that's why we've been committed to empowering you guys with more and more single cell multi-omic and sample type capabilities. And over a decade of single cell product development has now been condensed down into the product portfolio that we have at our present day, which is of course what you see here. There are 3 product families. We've got our Universal, Flex, and Epi, all under our Chromium single cell portfolio. So I'll begin with covering what's new for the Flex assay. We'll of course step through all the latest assay improvements, but from the get-go, I really want to highlight the key selling point for the Flex assay, and that's that samples are fixed at the sample preparation stage. So basically now you've decoupled sample preparation from running the actual assay. And it means you can build up a bank of samples, which you can batch process all at once. Obviously huge for a number of labs, as sample preparation is the most critical aspect of a single cell experiment, and now it can be done with a more standardized workflow and means no compromising cell quality by sort of rushing to get everything onto an instrument or into a workflow. As you see here, you take either whole tissue slices or your suspensions of single cells or single nuclei, and rather than having to prepare and run them straight away, you simply fix the samples using 4% PFA, and then you store them for later use. And then beyond the sample preparation advantages, this fixation also has the benefits of preserving biological states and fragile cell types. All right, so now you know the key benefit of using the Flex assay, and again, this allows you to fix samples and really decouples that sample preparation process from running the actual assay. We now want to cover what you can actually measure with the Flex assay, and the core measurement is obtaining whole transcriptome gene expression. However, you can also extend the menu of analytes to include cell surface protein expression, intracellular protein expression, and finally CRISPR guides. So in this instance, you can tell what the perturbation is that occurred in any given cell, which also get a whole transcriptome snapshot of the impact of that perturbation. So as you can see here, the Flex assay, it's quite versatile. So let's review what's new for the Flex assay. And we launched it over a year ago. It was very rapidly adopted by the single cell community, but just a couple of weeks ago, we overhauled the assay and introduced some new capabilities, which we'll now step through with these bullet points. And firstly, we actually changed the microfluidics that we use to encapsulate cells, which is what you see here in this little video. We call this the new GEM-X chip, or GEM-X microfluidics. Far more robust design, which is prone to less fluidic failures. But also importantly, this new microfluidic architecture also comes with the benefit of increasing cell throughput lane. So we've doubled the number of cells you can run in any given lane. So it used to be up to 10,000 cells per sample. Now it can be up to 20,000 cells per sample. And at the highest throughput, highest multiplexing capabilities of this kit, This is 320,000 cells per lane, or if you run all 8 lanes at once, that's over 2.5 million cells per individual chip. So you can see it really starts to supercharge those larger single cell projects where it's far more feasible than ever to scale up both cell and sample numbers. But also don't forget the assay will work on the extreme other end of the spectrum for just a single sample of just a few thousand cells too. And finally, we've also invested heavily in optimizing the upfront sample fixation process to ensure that you're maximizing sample recovery. So previous version of the kit, we required that you start with at least 300,000 cells into the actual sample fixation step. However, this meant there were a large number of people who couldn't use Flex. It was difficult to obtain that many cells from their sample types. So through careful buffer design and protocol changes, you can now start with as little as 25,000 cells into the actual fixation step. Beyond the bullet points of the last few slides, I want to cover more of the latest Flex capabilities. So I mentioned at the start, the assay is capable of measuring cell surface and intracellular proteins alongside whole transcriptome gene expression. So both protein tech and BioLegend cell oligo-labeled antibodies for this purpose. And then we've got updated demonstrated protocols which will guide you through the sample staining process. Next, you now have the ability to perform CRISPR screens in the context of the Flex assay. Again, I mentioned it before, but here you're detecting guide RNA molecules alongside whole transcriptome gene expression, so you can tell what the perturbation is that occurred in any given cell, but you also get whole transcriptome snapshot of the impact of that perturbation. With our new extended cell throughput with this new Genomics microfluidic device, you can start to do this now at a scale of thousands of guide RNA molecules and thousands of cells per guide RNA molecule. As you can see in our little data example here, where this was generated in-house, this is 7,000 guides screened in a Flex assay. We've also expanded our sample preparation options for the Flex assay to include fixation of whole blood samples. So previously, you actually need to process the whole blood to remove the red blood cells, and then you would fix that resulting suspension of single cells. But here you can simply take the whole blood as it is in that tube and you fix the whole blood, and then you deal with it later. You deplete the red blood cells later. Obviously, this is ideal for anyone working with blood, but in particular, if you're interested in those fragile cell types like neutrophils, they're better preserved with this sample fixation approach. So if you do work with blood, then I would highly recommend this approach. And finally, another exciting aspect of the assay is that it also works well on FFP samples. And there's no need to modify the actual single cell workflow when using FFP samples, just the actual upfront sample preparation, which allows you to go from this FFP block to a suspension of single cells. And we, of course, have demonstrated protocols which will walk you through how to do this. This is obviously significant for those of you who have FFP samples either already collected or if this is a primary sample format that you're used to working with. It really does open up your ability to generate single cell data from a whole bunch more samples. And this is obviously an excellent companion to any spatial measurements too. And this slide is just a quick data demonstration for the Flex assay where we've combined the new throughput capabilities of the Genomics microfluidics chip along with the ability to process FFP samples. So you see here that FFP samples from 8 distinct sample types were all processed in just a single reaction, which yielded about 320,000 cells in total. So not only have we easily scaled sample and cell numbers, but we've also returned some very high quality single cell data from FFP tissues. Moving on from our Flex assay, now I want to cover our next 2 assays being universal 3' gene expression and universal 5' gene expression. And we actually recently announced a pretty significant overhaul of these assays, and we hadn't really touched them in about 6 years. So we've taken apart the entire workflow from microfluidics, assay biochemistry, and even our analysis pipelines, and we put everything back together in a way that optimizes total assay performance. Before we dive into how these assays are actually overhauled, just a really quick refresher on why we have both a 3' and a 5' single cell assay. So on the left, we have our universal 3' gene expression assay. This captures the 3' end of polyadenylated transcripts. And using this kit, you can obviously measure whole transcriptome gene expression, and you can also extend that menu of analytes to include cell surface proteins. Now on the right, we have the universal 5' gene expression assay, which captures the 5' end polyadenylated transcripts. So with this assay, you can also measure whole transcriptome gene expression and capture cell surface protein information. Because we're now assaying the 5' ended transcripts, it's also possible to extract full-length paired VTJ information from T and B cell surface receptors, or TCRs or BCRs, if you will. It's also possible to extend the menu of analytes to include direct capture of guide RNA molecules from CRISPR screens. So to put it simply, if you're interested in whole transcriptome gene expression, then the majority of people are going to reach for the 3' assay on the left. But if you're also interested in immune repertoires or CRISPR screens alongside whole transcriptome gene expression, then you're going to reach for the assay on the right, the 5' assay. Besides the difference in analytes that you can measure, which are listed here on the slide, the workflows and assay specifications, they're basically identical. Anyway, like I said, we recently overhauled these 3' and 5' assays, so let's learn more about what this overhaul actually means. These bullet points I'm going to walk through highlight the key upgrades that we've made to both the 3' and the 5' kits. And firstly, we're actually changing the microfluidics that we use to encapsulate cells. This is the same design as the microfluidics I showed earlier for the Flex assay, so it's that GEM-X chip. This means that it's a more robust design, which is prone to less fluidic failures. It also reduces the runtime on the instrument from 20 minutes right down to just 6 minutes. And you might not think that's significant. It's a 2-day workflow. Who cares about 14 minutes? But for fragile cell types or even dead dying cells, the reduction in time spent on the chip prior to encapsulation can be really critical. We've increased the throughput of each channel by 2-fold, which means you can easily scale single cell studies to higher cell numbers. So you used to enable capture up to 10,000 cells per channel, and now it's up to 20,000 cells per channel. And then if you start multiplexing and superloading samples, you can be in the realm of hundreds of thousands of cells per chip. We've boosted the capture rate from 65 right up to 80% of cells from your sample. So this number describes a fraction of cells that are captured on the actual microfluidic chip. And it's a really critical number for those of you who have precious samples with really low cell numbers. So now you can be assured that you're going to capture a much higher proportion of the sample than ever before. 2-fold increase in cell throughput and increase in cell recovery also comes with a 2-fold reduction in the multiplet rate. So for every 1,000 cells, you're only getting a doublet rate of 0.4%. And finally, the biochemistry of the assay has been completely re-optimized to afford a higher sensitivity compared to the previous versions. And for everyone I talk with, it's really this metric that matters the most and closely followed by the cell throughput. So now we've covered those assay improvements, let's just see how they manifest themselves in actual data generated by examining assay sensitivity. So these examples are from the 3' assay above the top and 5' assay along the bottom. You can see there's a bunch of different sample types. Sequencing depth is on the x-axis and number of transcripts is detected is on the y-axis. The light blue line represents the previous versions of the assays, whilst the dark blue line is our new version of the technology. It's pretty obvious and striking. The new assays afford significant increases in sensitivity. And this isn't just an incremental increase or a small increase. This is a true step change in assay sensitivity. The next big advancement for the 3' and the 5' assay is the capability called on-chip multiplexing. And personally, I'm really excited about this. The field of single cell has been trying to reduce sample costs through multiplexing for a while now. And for those who are unsure what multiplexing is, rather than running one sample at a time, you simply combine multiple samples into a single reaction, and then later use informatics to assign any given cell back to a given sample. However, multiplexing has always involved adding some sort of oligo tag to each sample prior to pooling them, such as an antibody oligo or a lipid oligo conjugate. But upfront tagging of cells significantly complicates experiments. It adds extra sample preparation. It's generally only compatible for human or mouse. It requires lots of cells into the workflow, and overall, it can add significant experimental risk for some sample types. And my job is to have calls with people who are running single cell experiment. I can't say how many calls I've had where multiplexing is critical to bring down the cost of the experiment, but the risk that's added means it's failed and the whole experiment needs to be repeated. So that's why we're introducing on-chip multiplexing, where there's no tagging of samples whatsoever in order to multiplex them. Honestly, it's a really clever approach. 4 samples at a time are loaded onto the chip, where each sample occupies its own lane of the chip. Each sample is run on that own microfluidic lane, and then the barcodes assigned to cells are unique for each lane, so that's key. The output from all 4 lanes is simply pulled into a single well for processing into a single library, and then based on what barcodes are assigned to what cells, each cell can then easily be assigned back to a given lane on the chip, or if you like, back to its sample of origin. So here you can process 4 samples at once and collect up to 5,000 cells per sample. This approach obviously extremely robust because there's no upfront tagging of samples, and obviously a really great way to save money. So I really see the analytical utility of this kit is going to be great for pilot projects where traditionally pilot projects everyone wants to hash or multiplex to save a lot of money. but being new to single cell, don't have the experience to do that. So this is going to be a great way to instantly jump into single cell, multiplex samples in a really risk-free approach. I just want to cover some data from this new on-chip multiplexing capability. Here, 4 highly different samples were multiplexed into the same run. And what you see here is the combined output from around 20,000 cells. However, now you can see each sample displayed in its own plot. There are around 5,000 cells per sample. And remember, the great thing here is there was no upfront tagging of samples to do the multiplexing. Simply load each sample into their own unique lane of the multiplexing chip, and that's it. You're really just running them like they're singleplex reactions, but through that microfluidic device, we can multiplex them together. So hopefully you now inspired all the latest updates to our instrumentation, software, and assays. There's lots of new capabilities, and we're really trying to democratize single-cell sequencing for everyone and every sample. We do really want to be providing tools that maximize your data whilst minimize your costs. So I want to conclude my section on this slide. No matter what your single-cell project, we have the best single-cell solutions available. So if you're unsure about which single-cell assay will be best for your needs, then I'm always happy to have project discussions to help decide the most appropriate path to achieving your desired biological measurements, but of course, doing this at the best price possible. And I'm sure at this stage, in the presentation, you're thinking, hey, Geoff, it's been 20 minutes. And whilst you've mentioned minimizing costs a million times, you never actually highlighted how much money you can be saving. So with the final few minutes, I'm going to pass it over to my colleague, Marta from Millennium Science, who's going to go through assay instrument costs along with single cell promotions that they're running before the end of this year. So I'm happy to jump back on at the end and take any questions, but for now, I'm going to hand it over to Marta.
Thank you, Geoff. I will share my slides, share, and presentation mode. Yep, I hope that looks good. So welcome everyone, ho, ho, ho. Geoff said lots of interesting things and it really looks amazing, but actually how maximizing data and minimizing costs look in practice. So I would like to start just focusing on trying to maybe understand why actually we all need instruments, right? So if it was a seminar, I would ask you to raise your hand if you know what instrument I presented on this image. I know there's someone already that has actually used this instrument. So you might not be aware, this is the very preliminary version of the PCR thermal cycler with 3 different water baths with different temperatures that you will have to by hand move your PCR tubes, right? So actually, why it happened that the next step is really much more complicated instrument, which is basically present in every lab. And the next iteration of PCR thermocycler is qPCR. And in a very similar matter, this is the first chromium instrument from 10x Genomics many years ago. Some of you might remember the still recently operating Chromium controller and the newest family of Chromium X that Geoff mentioned. So what is happening here in every field is basically innovation is driving the change. And it is because we all want to have reproducible assays. We want precision, we want control of the conditions. And ultimately, what we are saving is not only money, but also time. Because, well, as we say, time is money, but it also includes your salaries and the time you spend repeating experiments or correcting any errors or optimizing things. This adds to the actual cost of your experiments. But actually, what does it mean with the cost savings? So just a quick reminder, 10x Genomics has 3 platforms, single cell chromium and also spatial xenium and CytAssist. But because this webinar is focused on chromium single cell, I just would like to show relative cost of the newest member of the family of the chromiums, Chromium XO. It costs only 1/4 of the Chromium X. And as Geoff mentioned, you can upgrade the instrument from XO to Chromium X to be able to access its full capabilities. However, we know it's not always fantastic in terms of funding in Australia and New Zealand. So there are also other options you can choose from. At Millennium Science, we have demo instruments for Chromium and Site Assist that if you're interested, there's a possibility of a free loan of such instrument to run your assays. If your project is getting started, it's getting quite large, we have an opportunity to bundle reagents and instruments together to reduce the costs. And lastly, we are working very closely with many core facilities across Australia and New Zealand who have these instruments and are able to run these assays as a service. So there is no need, if there is no capability to purchase the instrument, there is a plan C. So what about reagents, right? With reagents, it's actually very similar story to with the instruments. I don't know if you ever isolated a plasmid without the kit, but I did. And this protocol from 2020 is actually very much simplified. I think many people are just used to buying a kit and isolating plasmid. You forget that in the past, you might have to not only purchase the plasmid, which is ready, but also maybe produce your restriction enzymes and then purify them. But what is happening in the life science and in medical research, we want things to do much faster, much simpler, optimized, and in the best way possible. So when you're actually thinking about buying reagents from 10x Genomics, when you realize that actually in the kit itself is a set of extremely highly optimized reagents, including oligonucleotides, library preparation kits, ligases, RNase inhibitors, dynabeats, and all much more, just to create this extremely optimized set of reagents that work perfectly together in the setup of the Chromium instrument. So again, this is driven by innovation, just to have reproducibility, precision, overall control over your experiment, minimize the errors, minimize repeats that are needed, in the end, saving time, and this way, saving also costs. But there are also other ways that you can look at cost savings. So as Geoff mentioned, the newest edition of the GEMX kits allows you to do on-chip multiplexing, or in GEMX Flex, it's embedded also within the kit. But there are also other methods that Geoff mentioned, which are not fully supported by 10x, but we often discuss it during our calls with researchers. So you can do genetic multiplexing, which means if you have samples from different, for example, patients, and you have also genotyped them, you will know the location of particular SNPs, and you can recover that information from gene expression. And this way, you can separate the samples from particular patients. The next option is antibody-based approaches, so also known as cell hashing, where you label cells with antibodies conjugated with barcoded oligonucleotides. And then after running the assay, you can again separate the samples bioinformatically. Similarly, with lipid-based assays like multi-seq available from Merck, especially when no antibodies, no SNP, genetic multiplexing is available, lipid-based method can help you out. And lastly, core facilities usually have a capability of purchasing larger kits, which are usually much more cost-effective, and they can basically invoice you for the portion of the kit that they purchased. So if there are like certain parts of the kits that are purchased only in bulk, they can invoice you, for example, 1, 2, 4 reactions. So that's additional options. But talking in more details about costs, in terms of GEMX kits, in comparison to the previous iteration of the kit, so NextGem, these kits are already 10% less expensive. You can run 2 times more cells than on NextGem version of the kits. And there are these options to multiplexing that reduces the cost. So how does it look like? This is a plot that shows relative cost comparison of, let's say, GEMX Flex 4-reaction kits in comparison to GEMX Flex 4-plex. So 4 samples multiplexed into 1 run, total 16 reactions. And you can see as relative cost per sample, this is much cheaper, and it's getting extremely cheap the more samples you run. And also, lastly, I would like to mention that there's also the cost of sequencing. Oh, sorry, I forgot about that. This is just to emphasize which kits contain the option of multiplex, which is embedded within. And as you can see, they are much cheaper. Lastly, sequencing costs are additional costs. And when you are considering different ways of running single cells, you need to remember that sequencing is also included. That means there are no really true instrument-free assays if you need to do sequencing, unless you are planning to sequence on the vertical polyacrylamide gels, which I don't think you will. So there is always instrument involved. Just touching briefly on the data side, as Geoff mentioned, free software from 10x Genomics, free cloud, and also free tutorials and workshops. This is actually really important and something that if you learn the skill, it will actually save you cost in the future and time. As Lao Tzu said, give a man a fish, feed him for a day, teach a man to fish, feed him for a lifetime. So we are very much in supporting you to learn how to fish with 10x Genomics single cell and spatial data. But if that is not for you, there is always option of finding a collaborator or using bioinformatic analysis as a service in certain course. So with that, I would like to finish, and with Geoff, we will be taking questions. If you're interested in discussing how to minimize costs and use 10x Genomics, feel free to reach out to myself or Geoff. And thank you for your attention. I will stop sharing. And I'm not sure if we have any questions. We're on 3 minutes over time, Geoff.
Oh dear.
Feel free to put any questions in the chat or raise your hand. Otherwise, Geoff might start singing, so...
Better ask questions then.
Okay, Lee, let me allow you to talk. Lee, you can talk now, I think. Yeah. Yep, go ahead.
Can you hear me?
Yeah.
We can, Lee.
All right. Yeah, I'm just wondering about the multiplexing and the flex. Is it like a sample-based barcode system that's added before you pool them?
Yeah, great question. So actually, Marta, did you have a slide? I didn't have a slide on it, but so for gene expression flex, actually, maybe I do. We'll go back to that section. I don't have a picture in here, but basically the gene expression flex assay is a probe-based assay. So rather than directly capturing polyadenylated transcripts, we use oligonucleotide probes to bind to a transcript, and then we ultimately get a barcode and sequence probes rather than the transcript itself. So quite simply, to those probe molecules, we add a sample barcode. So it's inbuilt into the actual signal that you're going to sequence anyway, is the ability to multiplex samples. So if you like, you've got 4 samples in front of you. Each sample gets a set of probes to hybridize to the transcripts. Each sample has a unique probe sample index associated with it.
Yes.
Yeah, I get what you mean. So it's just for mouse and human at the minute, I'm guessing.
It is for just mouse and human. It has been expanded to other species. Typically, those species have high homology with either mouse or human. No one is, you can spike in custom probes. So we've got guidance on how to design your own probes to spike in, but that's generally for detecting species alongside mouse or human transcripts, like viral transcripts or bacterial transcripts. You know, expanding to plants, for instance, that would require synthesis of a whole new custom probe set. So
Yeah.
certainly feasible, but something that it has not quite.
In case of Lee, because Lee is actually working on plants, so in case of plants, you can, for you, would be then on-chip multiplexing and species agnostic, because the poly-A capture, so that would still work, right, Geoff?
Most definitely, that'd be what we'd recommend for sure. Would be on-chip multiplexing.
And the on-chip multiplexing, then I guess the same question, how do you distinguish between the different samples in that case?
Yeah, for sure. So each lane on the chip has a unique set of gel beads in that lane, and we know the barcodes on each of those unique sets of gel beads. So if you're like, we know all the barcodes that are in lanes 1, 2, 3, and 4. So when you run your sample and your transcripts get a barcode associated with it, you ultimately sequence that. So that barcode has 2 levels of information in it now. The first level is you combine all the same barcodes together, and that identifies, okay, all these transcripts came from the same single cell. And then we use that barcode to tell us which lane on the chip that cell was derived from. So it's, and then we can place that cell back in either sample 1, 2, 3, or 4. So again, it's inherent into the multiplex is inherent in the library that you're going to sequence anyway.
Right.
And,
Okay.
yeah. We have a question in the chat from Gavin. What is the upper limit on cell size for the GEMX?
It's a good question. So the microfluidic channel dimensions, I believe, and I might need to double check this out, sort of 50 micron by 50 microns square. That's what it used to be for our old microfluidic channels. I don't think they've changed for this one, but we'll triple check that. Either way, then that comes down to what's the upper cell size limits. Well, cells are squishy, so, and they change size in various buffers, so it's hard to say. I think cardiomyocytes, for instance, they're a larger cell type, multi-nucleated. We've seen people successfully run those on both the gene expression flex and the GEMX assays. But it really is probably sample type dependent, and probably honestly, if you've got really large cells, a great conversation to have with us ahead of time and see if there's a way we could perhaps screen those samples.
Are there any more questions? I think we don't have any more questions, and so thank you everyone for attending. Thanks, Geoff, for co-presenting. We wish everyone happy holidays and wonderful 2025 with lots of funding, great publications, and all the health and joy that you can get.
Take care, everyone. Have a good holiday.
Bye.